All right, microbiology class, welcome to the wonderful world of microbes. I think you're going to be surprised in this course how much you actually come to be interested in microorganisms. It's a part of biology that we don't really spend a lot of time thinking about until you get to this class. And I think you're going to be kind of fascinated by some of the examples that we find in here. So by definition, right, microbiology is part of biology. It's part of the study of life. But we're going to be looking at organisms that are really small. In fact, too small to be seen with the naked eye. So we're going to need to use magnification, usually a microscope, to do that. We have a wide variety of microorganisms. Some of these we'll spend more time on, things like bacteria and viruses. As you get further down in here, some of them are absolutely important in human health or causing human disease, but we'll spend most of our time on bacteria and viruses, right? And so just to give you an idea of some of this diversity here, these are those same groups that I had listed on the previous slide, things like bacteria and viruses. One of the neat things here is they're telling you the magnification. This is 15,500 times magnification in order to see that bacterium. I can't see my number on this bottom one here for the virus. Anyway, fungi, 400 times magnification. Algae, 500 times. The helminths are the worms. We'll see we include those in microbiology, even though some of them, like a tapeworm, for example, you could see that with the naked eye. But actually to be able to distinguish what species of worm that is, you would need something like a microscope because it's based on mouth parts and things like that that you can't see without a microscope. The archaea, the protozoans, etc. Now, what I want you to do for class on Thursday, right, when we actually meet via WebEx, is you'll see an assignment in Canvas asking you to fill out this table. So I want you to look for examples in all of those groups that I just mentioned. An example of a pathogen, right? A pathogen is just a microbe that causes disease. Usually you're looking for a human pathogen. If you're really excited about sea turtles and you want to find something there, that's okay, right? But we're looking for a pathogen, what the effect is, right? So what's the harm that it's causing to that host? Again, most of you will choose humans. And then maybe just some general information about that microbial group. The idea here is for you to get a little more familiar. These shouldn't just be like words out of your textbook, but to start to kind of get a working knowledge of what these different groups of microbes involve. So fill that out and we'll talk about it on Thursday. Okay? So microbes, we know they're small, right? That's part of the definition. And we saw from that list of 7 different categories that they're pretty diverse. And so one of the things I want you to realize is we have microbes in all 3 domains of life. Now, if you don't remember domains, right, think back to like middle school, high school biology, like kingdom, family, right, all that stuff. Domain is above kingdom. So these are really large groups of how humans organize life. And there's 3 domains of life. The bacteria, we have all of them are microscopic, right? The archaea, these used to actually be grouped with bacteria. They look very similar, but these tend to live in really extreme environments. Sometimes we call them extremophiles. They'll be in thermal vents and things like that. And then we have the eukaryotes, or the eukarya. And this is going to be things like fungi and animals and plants and algae, all that, okay? So microbes actually come from all 3 domains of life. Now, one thing that I did want to point out, you have an article to read called Retire the Prokaryote. So this is just to wet your whistle a little bit thinking about that. This is an old image of how we separated life. But what I want you to notice is we kept the prokaryotes kind of down here at the bottom of the tree. You'll see archaea and bacteria as prokaryotes. They don't have a nucleus. And then the eukaryotes up here, plants, fungi, animals, protists, which are things like your algae. Okay? This is no longer considered how life is related or what this tree looks like. And this maybe is an important lesson about science, right? In science, we're constantly learning new things, having to revise our viewpoints based on that data. And this is a much better look at how we think that life is related. And so you might notice things like the eukaryotes are actually more closely related to archaea than they are to bacteria. Anyway, you have an article to read called Retire the Prokaryote. It's making an argument that we should no longer even use this term that you probably learned in Gen Bio, and let's talk about that on Thursday as well. Okay, microbes are small. Again, just to give you an idea of what this looks like. A louse, right? This is haired lice. If you ever had lice or had to get them out of your children's hair, you can barely see them with the naked eye, or probably all of us have had bread mold growing on something in our refrigerator, right? That's macroscopic. That's visible with the human eye. Everything below this is going to be too small to see. Some of these things maybe you looked at in Gen Bio. Perhaps you looked in amoeba, or in anatomy class, we look at white blood cells and red blood cells. Okay. But notice, when we start getting into what we're going to spend a lot of time in this class looking at bacteria, they're much smaller, say, than a white blood cell, which looks pretty small on a microscope slide. So we're going to learn some new techniques and really hone our microscope skills. Now, there's many smaller microbes that we won't be able to see in this class. All of the viruses are going to be too small to see with our microscopes, and so we won't spend quite as much time looking at that. And then, of course, this drops even further, showing you the size of a protein, amino acids, specific atoms. We're not going there. Okay, so microbes are small. I will also say microbes are ubiquitous. Good word, right? If you ever get to hang out with your family again, you could use the term ubiquitous, which really just means that they're found everywhere, right? Every ecosystem that you can fathom, there's a lot of microbes there. Okay? And because they fit into so many different places, they've actually had a huge impact in shaping our planet. So here's just a few examples of things how microbes have been involved in shaping our planet. Photosynthesis, for example, right? So the early photosynthesizers, right, in our understanding of the history of Earth, were a group of microbes called the cyanobacteria, or the blue-green algae. And so they did photosynthesis, which, if you recall, the byproduct of photosynthesis is what gas? Oxygen. You got it. Good. And so they really played a big role in oxygenating the atmosphere and making it so other organisms could survive. In fact, today, microbes, things like bacteria and algae, account for about 70% of the photosynthesis on Earth. Therefore, 70% of the oxygen production on Earth. Now, it's kind of interesting if you think about it, because usually we think about things like the Amazon rainforest, right? Don't cut down the Amazon, it's the lungs of the planet. And I'm not suggesting you should cut down the Amazon, but 70% of the photosynthesis of the oxygen production is actually done by microbes. Okay, other things, decomposition. Bacteria and fungi are major decomposers. Without them, I know it's frustrating, right? They get to that loaf of bread just a little too soon, or your leftovers in the fridge. But think about it, without these organisms decomposing, we would just be up to our necks, we'd probably be walking on top of dead animals, piles of trash and waste, right? Decomposition, that breakdown, is super important. Not just so there's not dead stuff everywhere, but also so those nutrients can cycle, right? That they can be broken up into small parts that can get used again by other organisms. Greenhouse gas production. Microbes are huge producers of greenhouse gases. So things like during decomposition, for example, right, they're basically eating, they're doing cellular respiration. And so the byproduct of decomposition is going to be things like CO2 or methane, which are important greenhouse gases. Now, before you hate the microbes for that, recall, back in middle school science, right, the greenhouse effect is actually really important. If we didn't have gases in the atmosphere trapping heat in, Earth would actually be too cold for, say, humans to live on. And so the production of these greenhouse gases is really important. to a certain extent, right? If you start going above and beyond that, you're putting blankets on the earth, it's starting to get warm, people. Okay, symbiotic relationships. So there's so many super cool examples of symbiotic relationships that involve microbes. In fact, why don't you see if you can come up with another one. I'm going to give you 2 examples. So one of the classic examples of a symbiotic relationship is a coral reef, right? So coral is actually an animal. It's called a polyp, but it actually has algae that lives inside of it. So when corals have these bright colors, the coloration is actually coming from the algae. Now this relationship, the algae is doing photosynthesis and providing basically sugars, food, to the coral. The coral, the animal, is providing a nice warm, I shouldn't say warm, a nice habitat that feels safe to the algae, right? And so that's actually an example of a mutualistic relationship where both of those organisms are getting along. Probably the coolest examples of symbiotic relationships are mutualisms. The other one I'm thinking of is cows and their gut bacteria. So cows eat grass, right? Grass is just cellulose. Cellulose is a bunch of glucose molecules linked together, very much like starch. But the way the bond is formed between these glucose molecules, no vertebrate, nothing with a backbone, can actually break this bond. No vertebrate, nothing with a backbone, can digest cellulose. That's like all a cow consumes, the cellulose, is the cell walls of plants. So they can't digest it, but that's what they eat. In come the microbes, right? So in their 4 stomachs, they have these basically fermentation tanks where the microbes can break those bonds and then release the nutrients, the fuel, to go to the cow. So really cool relationship. Again, the cow's getting the energy, the microbe there is getting a home. Okay, I could go on and on about that. Pretty cool stuff. See if you can find another example of how a microbe and another organism have a symbiotic relationship. Okay, so lots of really important, say, like ecological roles of microbes. We're going to spend some time in this class as well thinking about how microbes are important to humans. Okay? So the first thing I usually tell students in this regard is that you have 10 times as many bacterial cells in your body as you have human cells. So this is kind of a joke, the human body, right? But like, what are you really if you have 10 times more bacterial cells than you do human cells? Okay, we'll spend some time thinking about that, right? We'll have time this semester to hash that one out. We also have managed to find very creative uses of microbes in order to produce food, right? So things like when you're baking bread, you're using yeast, that's a microbe. We use yeasts to ferment things like beer and wine, bacteria to make certain cheeses and yogurt, stuff like that, right? So humans have a very long relationship with microbes and using them for food. Biotechnology, right? So biotechnology is anytime you're using a living organism to produce a useful product. And there's lots of really neat examples. Probably the most famous forms of biotechnology with microbes have to do with genetic engineering. So actually going in and manipulating the DNA of an organism. That could mean moving DNA from one organism into another, or simply changing what's there. Some of the examples there, this picture is trying to show biofuels. So some researchers are trying to genetically modify different algae so that they can live in kind of tight quarters, maybe off of the CO2 gas coming off of something like a coal-fired power plant, and they can use that in sunlight for photosynthesis. They're trying to get them to produce higher levels of oils so they could be used for fuel, things like that. We have genetically modified bacteria, for example, by inserting human genes. We have inserted human genes into the E. coli bacterium in order to get E. coli to produce human insulin. This is where a diabetic, when they have to give themselves an injection of insulin, they have a little vial. That is made by bacteria, but it's actually a human protein that's produced, and this is the result of genetic engineering. So that's a pretty cool example of biotechnology. Last one, bioremediation. So things like oil spills. We've found certain microbes that can help clean up those oil spills, actually digest petrochemicals, mine waste, things like that. And so we can use them to clean up problems. I'll also include, it's not really a use of a microorganism, but one of the big reasons that humans are interested in microbes, of course, is infectious disease. Now, don't start writing this all down. You don't have to memorize these percentages. A couple things you should know, right, is that a pathogen, by definition, is just a microbe that does harm. What I want you to gather from this is an image out of an old textbook, is that there are thousands of different microbes that actually cause disease. And these numbers, they shouldn't be that surprising, right? If we have 9 billion people on Earth, think about it. We've all had a cold like every year, right? So it's not surprising that there'd be 10 billion infections or 12 million deaths a year from infections, right? This is kind of a breakdown, again, not to memorize, but kind of understand where those illnesses fall. So a lot of respiratory illnesses, things like pneumonia, influenza, we could add coronavirus in there as well. AIDS is a large portion. This has got to be deaths then. Diarrheal diseases. People still die from diarrhea. They get dehydrated. In a lot of third world countries that can be lethal. And some other ones that we'll talk about here as well. We'll get into parasites in a future chapter. A parasite just lives in or on a host. Basically, as long as a microbe's not killing you, if it's trying to live in or on you, you could consider that a parasitic relationship, right? It's taking resources from you. So you can be both a pathogen and a parasite, right? Typically parasites are doing harm. This I did think was interesting. This is an image out of your textbook showing causes of death. And it's just a neat comparison of the U.S. versus the world. Now, this is all kind of up in the air right now, right, with COVID-19. But notice what they're showing you here in the U.S. Heart disease and cancer are major killers. We don't hit an infectious disease. This is chronic respiratory, so things like emphysema. Accidents, stroke, Alzheimer's, diabetes. We don't hit an infectious disease being a major cause of death until we hit influenza and pneumonia. So they've put those two together because oftentimes people with influenza, what actually kills them is pneumonia that follows that. So anyway, this is like the number 8 cause of death in the U.S. So big picture then, what we're looking at is if you look at deaths in the world, again, heart disease is in fact number 1. Then it goes stroke, COPD, this is again kind of like emphysema stuff. But we hit those respiratory infections, right? The influenza, pneumonia, stuff like that. Then we're dropping down into some cancers, lung cancer, HIV, AIDS, diarrhea, which don't even show up on the US map. So anyway, kind of an interesting look at how developed nations versus developing nations in the world might be different in regards to infectious disease. Tangent though, it's tough for you. You can look in your book if you want to see these numbers. This is 175,000, this line. We're already with COVID-19, I'm recording this on August 10th. We're already at 160,000. So this year, we're going to get these respiratory diseases bumping up in the US to the number 3 cause of death. And we'll see where that goes. But that makes us look much more like a developing nation. We can talk about that. Okay. Okay. If you have any questions on any of this, it's all kind of introductory, hopefully just kind of get you thinking about microbes. Write them down. We can talk on Thursday. Last thing I want to talk about here before we wrap this up, right, is a little bit of history. So you can pull this PowerPoint if you're interested in. The Howard Hughes Medical Institute has a website called BioInteractive that has a little video talking about Antonie van Leeuwenhoek, who is the first person I want to talk about as far as the history of microbiology. So again, you don't need to know these guys in excruciating detail, but to kind of have an idea of the time frame. You don't have to memorize dates, but you should have some kind of idea of when these things were happening as far as the history of the world in microbiology. So Antoine van Leeuwenhoek, he's a Dutch linen merchant, and so he's actually interested in thread counts and fabrics. And so he starts grinding these glasses, these little magnifying glasses, so that he can see these fabrics better. But as he's getting better and better at developing these, he puts a drop of water off of his, it's raining, he grabs a drop of water off the roof and looks. And is shocked to find things seemingly living in there. He refers to them as animalcules, but he's basically the first person to see this microbial world. So you're talking late 1600s, early 1700s. Before that, people had no idea that anything existed that was smaller than we could see with the naked eye. And we could have a whole conversation about what that means as far as why we need science to improve our understanding of the world. Right? Now, when we say he invented the microscope, this is not like the microscope you have in class. You actually are going to have a much more advanced microscope. These little lenses, they were not amazing, but they were just enough to let him see into this microbial world. Okay, Pasteur, he's like the father of microbiology, so this is an important one to know. Unfortunately, you're already familiar with the name. He developed pasteurization, which is a way to kind of like flash heat things like milk and juice to kill pathogenic microbes, so microbes that could make you sick. So you're probably already familiar with his name. Now notice, he is basically in the 1800s. So it's really late 1800s when he starts doing a lot of this really important work. Okay, so a couple of the key things that he figured out. He showed that microbes caused fermentation and spoilage. He actually got interested in this. He got brought in by the wine industry, which was trying to figure out what was going on with some of their wines. He disproved spontaneous generation of microorganisms. So this was the theory, I shouldn't even say theory, this was the belief that people had called spontaneous generation, where like mice are crawling out of this bin of grain. Mice just appeared, right? Or like flies start, like maggots start coming out of some meat that you left out overnight, right? That was spontaneous generation. People didn't realize that a fly was laying an egg and a maggot was coming out. So he was able to disprove this theory of spontaneous generation of microbes, right? Like, man, where did that bacteria on my bread come from? So basically what he did was he made a broth. We'll work with some broth here this semester. And he boiled it to make it sterile, so it would have killed any microbe in there. And he actually made this special glassware with this long neck, right, so air can move in and out. But apparently, microbes can't. When that neck is intact, that broth would basically not get gross, right? But he came back later and broke the neck, right, and within hours or days, microbes are entering the broth and growth is occurring. So he's able to show that even though we can't see them, microbes are in the air around us and they're able to spoil things. Right? So he's able to disprove this idea of spontaneous generation. Developed pasteurization, and he also developed this germ theory of disease. That basically is saying that it's actually microbes, germs, causing disease, not evil spirits or humors. Like, we don't need to believe people. There's bacteria, right, making them sick. This guy is kind of a big deal. Okay, Robert Koch, you'll notice him and Pasteur really overlap a lot. So they're working at the same time. And Pasteur is really famous for a couple things. So he developed a lot of pure culture methods that we still use today. So in this class, when you come to lab and we're trying to grow E. coli in pure culture, like keeping anything else from growing there, we're going to be using some of the same recipes that Koch developed in order to have broths and augers and things that keep petri dishes, things that keep bacteria growing. Anyway, He also was able to identify the cause of some really important diseases in the late 1800s. And he did this through what are called Koch's postulates. And so Pasteur was like, hey, microbes cause disease. Koch is actually able to sit down and sequentially decide that yes, this is in fact true, and figured out which bacteria cause anthrax and tuberculosis and cholera. So this is how Koch's postulates work, right? Koch says, okay, you have organisms with the same set of symptoms, right? They clearly have the same disease. Let's figure out what's going on with them. So we are going to take a sample from them, right? And in all of the sick organisms, we're finding this like green blob, this suspected agent, and we're not seeing any of that in the healthy organisms, right? So his first step is that suspected agent has to be in all the ill animals, but not the healthy animals. Now, we are going to grow that organism, the microbe, in isolation. We're going to separate it from anything else, and you'll learn these techniques this semester for growing just that bad guy, okay? So most people at that point would be like, sweet, we were able to pull a microorganism out of all the sick animals and none of the healthy animals. That must be what's causing it. That is not how Koch works. Koch goes on to say, well, we're not really sure yet, so let's do this. Let's take that organism that we suspect and let's inject it into this healthy mouse and see if it dies. Actually used a lot of guinea pigs, hence the term guinea pig. We would expect the same disease to occur in these organisms when they're given this inoculation or this injection. And most people would say, oh, okay, cool. Koch says, no. Now you're going to take that diseased organism, take another sample, and make sure that what he's calling a causative agent, the organism, is there. So we've got 4 steps to Koch's postulates. The causative agent is in all of the diseased animals, none of the healthy animals. We grow it in what we call pure culture, isolated. It's the only thing there. And then we inject it into a healthy organism, causing the same set of signs and symptoms, right, the same disease. And then we're able to re-isolate that same organism. And then Koch says, that's what caused cholera, or that's what caused anthrax. So Koch is like super meticulous at figuring these things out. So, really important guy in history. Okay, last couple, Semmelweis. In the 1850s, he decides that hand washing is a good idea. So, I think your book goes into it. There's this whole story. All these women are dying in childbirth. They call it like childbirth fever. But what was happening is the medical students are doing autopsies on these women that die in childbirth, and then they're going to deliver the next baby. And they're delivering the bacteria with them that then infects that mother, and she dies the next day. And they're doing the autopsy on her, and then, right? And so, anyway, Semmelweis says, wash your hands. So he's going to be a major theme this semester, right? Because we're washing our hands. Joseph Listerine, okay? He basically comes up with what we call these aseptic surgical techniques. So he decides, hey, if I'm going to slice someone open, if there's bacteria or these microbes on the surface of their skin, that could actually infect them and become problematic. And so he's famous for these aseptic surgical techniques. Again, late 1800s. So kind of the same time that Pasteur and Koch are really trying to figure out what microbes are causing what. Semmelweis, again, we had very little understanding of what these microbes were in the 1850s. He just says, oh, hey, if you wash your hands, mom doesn't die. Okay, and so that worked out nicely as well. Okay, for Thursday, you'll see that there's a few different assignments, a couple of which I've talked about. You do have a movie and a short article to read as well. You just need to submit like a paragraph response, but we can talk about that on Thursday when we meet on WebEx. And, when you're reading chapter 1, one of the things we're really going to focus in on Thursday, because I've covered a big portion of the chapter right here, one of the things I want to focus on in our time together is the macromolecules. So really spend some time reviewing from Gen Bio your proteins, carbohydrates, lipids, nucleic acids, and we'll spend some more time on those in our WebEx meeting. Okay, I will see you then. Have a great day.